So in the last class we just began getting familiar with C. elegans as a model, right. So we stopped at this slide, like sex determination is done by XX means hermaphrodite, X0 is male, right. So we also saw the advantage of this because when it is easy to set up a cross as well as it is easy to set up self cross. Self cross is simply cloning a hermaphrodite. So one of the big advantage of that is bringing recessive alleles to homozygosity and therefore seeing the phenotype is relatively straightforward. If you pick from a mating cross, if you pick the hermaphrodites and clone them, and 1/4 of their progeny will be homozygous. So right away you will be able to see the phenotype. So that is one of the big advantages of this system. So let us move on. So how to make use of this to discover new genes. So before that, one more set of experiments which we are already familiar with. We have already learnt about fate mapping, right, in that lineage tracing. So in that discussion what we did was we focused on finding the origin, the original cell from which an entire set of descendants, like let us say a tissue, from what embryonic primordial cells a given tissue is derived is what we learnt there. So here we are going to do a little bit more advanced way of thinking about it, so it will become clear as we go through. So before that, so this shows you the early embryonic lineage, only a part of it, focusing on the germline just for clarity. So the zygote with one nucleus, we call it as the P0. So that divides into two cells, one cell we call AB and another one we call P1. And if you look at the horizontal line, you find the one on the left little longer from this vertical one compared to the other one. That is not random, it is intentionally drawn that way to indicate that the cell on the left, that is AB, is a larger cell compared to P1. So therefore there is an asymmetry, and therefore when I say this is anterior and this is posterior, it is not difficult, right. The one where we have the smaller cell we call posterior and we call that P1. So some of the cytoplasmic components get segregated during this process to the posterior cytoplasm of P0, and therefore they are inherited only in P1 and not in AB, and vice versa as well. Okay, so that there itself it happens, but today that is not our focus. So then the next division and AB divides to give ABA, ABP. So this small letter A indicates anterior daughter of AB, P means posterior daughter of AB, and this might go on AB, A, P, 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 PA, like that it will go. Each letter indicates one generation. And P1 divides into two cells, one is called EMS, another one is called P2. So these are same nomenclature as you saw in Ascaris embryo that was observed by Theodore Boveri in his experiments where he found chromosome diminution, if you remember, right. And then P2 divides C and P3 divides D. I said partial because I am not drawing all the descendants of these blue colored cells, okay. So only one we followed. So this is a partial lineage. This is just to familiarize you with how this lineage is shown in a diagrammatic way, okay. So we call this as a lineage tree. So we have this kind of lineage tree for all the 959 cells that make up the adult body. So like Ascaris, here again the embryonic cell division is invariant. It is exactly in the same pattern, only it divides in embryo after embryo, like automobiles produced in an assembly line. There are no variations at this stage. So later in the adult stage you might find some worms little bigger, some worms little smaller, like longer or shorter, or some may be little obese compared to another one that may be leaner. That is due to changes in cell size itself. Okay. So, the embryonic cleavage pattern is invariant, by contrast our cell divisions are not invariant. So, this invariant pattern allowed John Sulston, he passed away I think last year or the year before. So, he shared a Nobel Prize with Sidney Brenner and Bob Horwitz. So, John Sulston did this up to the adult stage. Okay, the early embryonic lineage he completed it, and when he did that, he accidentally discovered evidence for apoptosis. That is, some cells born in a certain lineage always died. So, that one could easily reason out, when you repeat multiple times, then you reason out that that must be developmentally programmed to die. So, that is how they obtained the first conclusive evidence that apoptosis exists, and that is what and then Bob Horwitz figured out the genetic mechanisms, okay, what are the genes involved and how do they function in executing apoptotic program, and that is the reason he shared the Nobel Prize. So, John Sulston got it for complete lineage of an organism for the first time, and Sidney Brenner got it because he started this model system, recognized its importance, all of that. So, why are we interested in lineage analysis is coming in the next slide. So, you can go ahead and kill a specific cell without affecting the other cells, okay, at different stages during embryogenesis, and then monitor the consequences. And when you do that, you can define a phenotypic defect of loss of function of a gene, meaning a gene function at a single cell resolution. Okay. In this particular cell, in this particular time, this gene must be functioning, then only this whole thing develops. Like that, at single cell resolution you can define a phenotype, okay, instead of telling this in the absence of this gene the plant will be dwarf. So, why is it dwarf? So, you go back and see, okay, this tissue does not develop normal, the proliferation is less, then you go back and find out why is that tissue having less cells, and go back like that. That is what we call as precise phenotyping, and that in this context can be taken all the way up to a single cell. So, that is what we call a single cell resolution. Second, are there potential cell interaction? Okay, if one cell is not present, like you killed it by laser ablation, then what happens to the fate of other neighboring cells? Are they autonomously developing, or they were dependent on this cell that you killed? So, you can discover potential cell interactions. Then you can also find out the developmental potentials of a single cell. For example, a given cell might develop into muscle when the normal neighbor is present, and when the normal neighbor is not there, like normal neighbor could have induced, remember the induction that we learned when we learned the 3 germ layers, and if that is not there, what is the default potential of this cell? Okay. Like for example, a genetic example that we have already learned is this testis determining factor. In the Y chromosome you get rid of that gene, then normally that embryo is going to develop into a female. So, then you realize the developmental potential normal in human is actually becoming a female. So, that is the default state. So, you can find out what is the default state of that cell. Then when does that cell get committed to a particular developmental path? Okay, so, where like for example, if a neighbor induces a particular cell to become muscle, when does it induce? Like how early should I ablate that? So, these are the things you can discover simply by doing laser ablation. Okay, so, now let me tell you what is laser ablation. Laser ablation is basically, So, obviously, the ones that we are seeing here, these are microscopic. Okay. So, if you have to kill a specific cell, you are not going to be able to take a scalpel blade and go and surgically remove it, okay, because these are too tiny. So, the way it is done is you mount it on microscope slide and you under the microscope using the microscope objective lens itself, you shoot a micro beam of laser, and the beam size is so small that it can specifically kill the nucleus of a given cell. Okay, so, that is what is laser ablation, and this technique is widely used in many model systems, and people also use a variation of this called laser capture microscopy, where you can specifically pinch off a cell from a tissue and then you isolate RNA, protein or whatever you want from single cell and then study that. That is called LCM. So, that is laser ablation. So, this is what is used here for this analysis. Let us see an example how all these four can actually be learnt through lineage tracing with the help of laser ablation. Okay. So, we are going to learn this as well as some of the genetic concepts in the context of making a particular organ. Okay. So, let us say our question is organogenesis, how an organ develops, what are the signaling pathways involved, when does it get committed, all aspects of an organogenesis. So, the organ about which we know the most of any organism is the C. elegans vulva, okay, the apparatus through which it lays out the embryos or mates with a male to get sperm. Okay. So, that is the organ we have learnt the most. That is the organ in which we first found out that RAS signaling is not the concept of proto-oncogene, that is an obsolete concept. Okay. So, people thought that there are certain genes that are waiting to be activated, therefore, they can cause cancer. That is not how cancer happens. Cancer happens because a normal gene that should be shut off at a certain point is not shut off, or a gene that should not be expressed in a certain context gets expressed, and as a result they end up stimulating proliferation. Okay. So, people realize RAS is not an oncogene that is required for normal development, and in its absence development does not happen. So, that kind of RAS signaling, then there are other signaling like Notch delta. So, they have all been discovered while solving the problem of how vulva develops. Okay. So, therefore, we are going to use vulval development is our example where we are going to learn basic genetics, and we begin with the lineage analysis itself. So, if that is what we are going to use as an example, then we need to familiarize ourselves with vulva. This is a section of the worm. Remember yesterday I showed you the full worm. So, this is roughly the middle part of the worm that you are seeing here. Okay. So, this is roughly the middle of the worm, and this is the pore like organ through which the embryos come out. Okay. So, this is the opening of the uterus. So, you have two gonadal arms that connect to a central uterus where you have embryos, and the embryos come out through this, and these are muscles that help in the contraction to push the embryo out. So, these are called these are the vulval muscles, this is uterine muscle. You need not worry about it, this is just to give you a pictorial view of how the C. elegans vulva looks like. And these are ventral cord cells, we need not worry about it. So, these are neurons connected to the vulval action. So, these neurons are shown here. So, our focus is only these, okay, the vulval muscle sets and vulva itself. So, you can get a very good view of all parts of the worm in 3D in different sections, head to tail, whatever you want to see, all of it if you go to this website called wormatlas.org. Okay. You want to see cartoon view, or you want to see scanning electron micrograph view, or you want to see transmission electron micrograph, you can get all of it for any part of the worm. Okay. It is available there, wormatlas.org. So, this is looking really at the pore using SEM image that shows you this is the opening through which the embryo comes, and you see these many structures they finally make that happen. and this is using GFP that is expressed only in this particular tissue. Okay, another example of tissue specific expression. Remember the crystalline protein coupled with GFP expressed only in the eye of the zebrafish. So, similarly here again you have a tissue specific expression here. So, this is vulva. Now, our question is how to make a vulva. Okay. So, this is the lineage diagram for vulva. So, in the very early larval stage you have 6 cells, okay. These we call them PNPs, okay. P3, 4, 5, 6, 7, 8. So, these 6 cells are the ones that are eventually going to make that entire vulva, and they go through this lineage shown here to make this, okay. And we call this cell whatever it is going to do, based on that we call this as having acquired primary fate, and the 2 adjacent ones we call them having occurred secondary fate. That is because certain structures come from cell that has got a secondary fate, and then you have these 3 cells that have the tertiary fate. So, from the early larval stage then they get these 3 different fates, and based on those fates then they end up following this lineage. Okay, like for example, a cell with the tertiary fate where the phenotype is still not visible is not going to provide these cells to make the vulva, only the primary fate cell can do that, and so on. So, now so that is summary of after all the experiments. So, we are going to do simple experiments. So, we know that these 6 cells are the precursors, okay, PNPs. Now, in an intact animal you follow the lineage of any one of the 6, then you end up finding P3P is always giving rise to tertiary fate, and P4 and so on. P6 always gives primary fate, and based on that you say it is invariant pattern, okay. And one larvae to another larvae there is no difference, and therefore, these cells do exactly this, and that is an invariant pattern. So, that is the conclusion when you observe them again and again. Now, let us say you go and ablate all gonadal cells. We did not look at the gonadal cells, okay. So, here for example, this is one of the gonadal cell here. So, the somatic gonad is on top of these cells, okay. So, you go and destroy all of them, then what happens? All these 6 cells they only have the tertiary fate. So, they do not have this primary and secondary fates. Now, you leave out the gonad intact like the rest of the gonadal cells, but only one cell called the anchor cell you ablate it, then you get the same effect. None of these PNPs acquired the secondary or primary fate. Then you do one more experiment where you ablate all gonadal cells leaving anchor cell, then you end up finding it is like wild type. So, these 3 put together tell you that it is the anchor cell that induces these cells to acquire their fate. Primarily the secondary and primary fate, for these 6 cells to acquire that you need the anchor cell. So, this is how you work out to a single cell, the anchor cell is the one that induces the primary and secondary fate. Now, you want to find out, so this gave you cell cell interaction, right, evidence for cell cell interaction. So, now you want to find out what are the different things each one of these 6 can make. Now, you go ahead and kill that P6P, and now in its position there is no cell, and the neighboring cells P5 or P7, one of them purely by random becomes primary fate. So, that tells these cells have developmental potential to become primary as well as secondary. Okay. So, P and P's are multipotent. So, that is the conclusion you come to. So, now you get rid of all anchor cells and all the PNPs leaving only one alive. That gives you the default state. When the induction is not there and neighbors are not there, what happens to a given cell? So, it occurs the tertiary fate. So, this tells you the tertiary fate is the default or the ground state. But given the induction, they can become primary or secondary. So, essentially any one of these 6 cells can acquire any one of the 3 fates depending on the context. Okay. So, what are the neighbors is the inducing signal there. So, all of this could be learned by simple laser ablation, a microscope and slide, and that is it. No restriction enzymes, no antibodies, no western, northern, immunostaining, immunohybridization or NGS or anything. Alright, so that is to that is an example of how lineage analysis is useful. Next is we are going to do a genetic screen. Okay, why do you do a genetic screen and what do you learn from it and how to do it, that we are going to learn using an example here. So, here our goal is how to make vulva, right. So, before beginning I want to tell you one really fundamental obvious thing, but we do not often think conscious about it, which is when you look at the DNA sequence, the order in which the 4 nitrogenous bases occur hold biological information, but chemically they are either purine or pyrimidine. There is no uniqueness in terms of chemistry for any given sequence. Okay. So, as a result, anything that is going to chemically disrupt DNA is not going to be sequence specific. Okay. So, we need to remember this. Like for example, when Morgan's group used X-ray to mutagenesis, they can't target, I want to start with chromosome 1 left side first 100 base pairs. You can't do like that. Okay. So, we need to remember this. So, therefore, when you are going to mutagenize using whatever mutagen, you are going to randomly disrupt DNA. Okay. So, depending on the strength of the mutagenesis that you are doing, okay, like for example, if you are using a chemical like ethyl methane sulfonate shown here, okay. So, shown here EMS, depending on the concentration of EMS you are going to use and for how long you are going to expose your organism to it, by controlling both of that you can mutate such that only in a given organism one part, a small part of the DNA is damaged, but not the whole of it. If you take lot of EMS and put the worms there for a long time, they are going to be dead because you are going to make large scale disruption on the DNA. So, here optimize the concentration such that you will probably create one lesion on the DNA somewhere in one chromosome or the other, not multiple mutations. Okay. So, that is how you do it, and after doing it in a given organism you can't predict where the damage is going to be on the DNA. So, therefore, what you do is you follow the effect of mutagen by following the phenotype. Okay. Is it going to make wrinkled seed or round seed? So, that is always the readout here. Okay. So, therefore, what will be our readout? Is the worm able to lay embryo or not after treating with mutagen? So, that is what we are going to look at here. So, we already saw this self fertilization allows quick recovery of recessive mutations, and these are listing some of the advantages, the first 2 bullets, advantages of using C. elegans as a model. The second one tells you the number of chromosomes that screened is twice the number of F1 progeny. So, what that means is, if you are going to take, let us say, hermaphrodites, okay, you wash a plate a few 10,000s of hermaphrodites, young adult, and soak it in a solution containing EMS, you are likely to have sperm, in some cases sperm where mutation has happened, some cases oocytes where the mutation has happened. Okay. So, therefore, after mutagenesis treatment with the mutagen, when I am going to clone the worms, I am going to look at the progeny, you are actually having a chance to screen both effect on oocyte or sperm. It could be either one, but you will be able to screen for both. Contrast this with an organism where you have male and female separately. If I am going to take the female and mutagenize, and in the progeny I am only having mutations coming through one gamete if there are no mutations in the other one. So, that is the advantage here. So, if I am going to screen 10,000 F1, what it means is I am actually screening 20,000 chromosome sets, right, because sperm and egg come from the same individual. And usually it is enough if you screen about 5,000 worms, like 10,000 gametes, to find loss of function in any given gene, given its genome size and the fact that you have the ability to screen twice the number of chromosome sets per individual organism. Usually screening about 5,000 worms is enough to get a loss of function like lethal or sterile, those kind of phenotypes. We will get into complex genetics where you need to screen like 1,000,000 or more, and even then you may not get. So, that is a different story. But to pluck the low hanging fruits, mutagenize and look at about 5,000 worms, that is good enough. It is very readily doable. Okay. So, usually 50 millimolar EMS is what is used, and they usually soak it about for a few hours, no more than that. Then they wash off the EMS and then put on plates, allow them to feed on E. coli, and then you collect embryos from them. Okay, so this is an actual screen here. So, you take the worms wild type, okay, for a given gene involved in vulval development. Since this worm has functional vulva, we will assume that gene is wild type, plus over plus, okay, it is diploid, right. Then after you treat with EMS and then you allow it to self, meaning the hermaphrodites are going to give their own progeny using their own sperm and egg. Now, let us say your gene of interest is mutated in one of the two allelic chromosomes, and we call that as M, and the other one unlikely that exactly the same locus got mutated in that too, the probability is really low. So, therefore, we write it as M over plus. Now, F1 worms, if you clone in F2, you will get 25% of the worms not laying any embryos, and here they are going to be bag of initially embryos, like they are not laying embryos out. So, the worm gets filled with embryos, and the embryonic development unless like our context where you have placental connection to the mother, and if mother is not feeding, the embryo is not going to develop, right. Here it is happening inside an eggshell. So, it is all packed already, the food is packed and given and the journey continues, okay, inside a eggshell. So, the egg is not going to care whether it is inside the mother or it is outside, it is going to undergo the embryonic development and become a hatch and come out as a larvae. And when you are going to have a worm filled with worms inside, even in 1 in 10,000 that worm will stand out. So, that is another important aspect of how to design a screen. The phenotype that you are going to look for should be very obvious compared to the background wild type. So, then it is very easy to score, very easy to identify even a very rare mutagenic event. If your screen, the readout that you are going to plan is such that you can readily observe it even in the midst of thousands of wild type progeny. That is an important thing in genetic screen. So, here this was an easy thing, you are going to look for, you have a plate, let us say I put lot of F1s, like let us say 10,000 F1s, I am looking at the progeny, I may not need even more than 2 seconds to look at a plate. The moment I look at the plate, a bag of worm, I will immediately know. So, then you pick that. So, that was the result of the screen. So, what did they get? They got about 100 mutants in the initial screen where there was problem in laying embryos. And when they decided to look closely at it, they found some of them had multiple vulval primordium, as if it is trying to make more than one vulva, and in some of them there is no vulva, okay, they could already classify into 2. Then it is possible that you isolated multiple loss of function alleles for a given gene. So, this 100 mutant alleles, like you have 100 groups of worms where there is a problem with making vulva. This does not mean you have actually identified 100 genes involved in it. Is this clear? Some of them, maybe you got 10 alleles for one gene, and maybe in another gene you got only one allele. So, it is all possible. So, how do we identify how many genes did we actually mutagenize? So, and that is where you do complementation. So, have you heard of complementation earlier? Right, you do complementation whenever you clone in PUC18 derived vector like blue script or whatever, blue colony or white colony, that is a complementation, alpha complementation, you do that there. So, what actually you are doing is you take one of these 100, okay, and then mate it with another group, okay, and if mutation is on the same gene, then they will be homozygous for the loss of function of that particular gene. So, they will be vulvaless. But one is mutant in gene A required for vulval development, another one is mutant in gene B required for vulval development. If you have A m over A plus and B m over B plus, then you have wild type copy for both of them in the trans hetero, we call that as trans hetero. So, that will be wild type. So, that this is what is complementation. So, if they complement, then they are two different genes. If they do not, then they are on the same gene. So, therefore, the mutant, each mutant having a phenotype, we call it as an allele, because whatever be that locus, there is a wild type allele and you got a new mutant allele. So, there is no problem in calling it as an allele, but after doing complementation, some alleles will group into one group, another one will group into another group. So, you actually call it as complementation groups. So, each complementation group is essentially mutations for a given gene, alleles of one gene. So, this is how the scientists came down to 20 different genes. So, one mutagenesis screen, then complementation, now we have 20 genes in vulva development. Okay. All right. So, now let us go further. Okay, 20 different genes involved in vulval development, but then we are working not on a difficult organism. So, here we know invariant lineage, we know laser ablation, and we thought that we can define phenotype to single cell resolution, right. So, let us do that. Can I now find out in each one of the 20 where exactly is the problem to start with during the embryonic or larval development. So, that is the analysis here, detailed observation of mutants instead of simply looking at the plate where you have lot of worms in one plate and then under a dissecting microscope all you can say is, oh yeah, this does not have vulva, this has vulva. So, the one that does not have vulva, where exactly the problem is, or during the larval development when do I first see a difference from the wild type. Okay, so, that is what we call as detailed observation. So, Nomaskia Optics, are you familiar with it? Okay, anyway, I will briefly tell you what it is. So, when you have a thin transparent specimen, let us say all of you must have done some practicals in botany in school where you did a cross section of a stem or a root and put on a microscope and saw, and then you stained with something and saw, right, you have done it. So, now if I am going to take a thin section and allow light to go through, which we call bright field, many internal structures will not be very readily visible. If they are all going to be transparent and allow light to pass through, it will not be readily visible. So, you will not get you will not be able to visualize some of the internal structures, and you may not get a 3-dimensional perspective to the image when you do that sort of a microscopy. So, a person by name Nomaskia, he came up with a method. Okay, so, this uses several lenses and optical components in the microscope, such that the differences in the refractive indices of the different cellular components, let us say the nucleoplasm has a different refractive index compared to the cytoplasm or the nuclear membrane or mitochondria and so on. So, by exploiting the differences in the refractive indices, he managed to create a 3-dimensional view of an object, and that is what we call as differential interference contrast microscopy, DIC, or Nomaskia, okay, in honor of the person who came up with this method. And that is what that is the image you are seeing. When I first showed you worm, that is a DIC image. Okay. So, every one of those images were all DIC images. So, here there is no cutting, staining, nothing, it is just regular light microscopy with no staining, but by exploiting the differences in the refractive index, you get subcellular detail as well as 3-dimensional perspective of the object. So, using that, you go ahead and examine what happens to these 6 cells. Okay. So, wild type we already know, invariant pattern, then one of those 20, okay, Lin26. Okay. So, in that the phenotype was vulvaless, that is what initially we saw. Now, when you observe, you find there are no PNPs, none of the 6 were present. That tells you the wild type function of Lin26 is required for PNP production. So, you discovered a gene that is required for the PNP formation to start with. Then another one you look at it, Lin3, obviously 2 different complementation groups. Again the phenotype is the same, no vulva, but when you look at when exactly the problem started, then you find the PNPs do not seem to get the acquired fate. Okay, so, they all have the default thing, showing conclusively that Lin3 is required for the induced fate. Okay. In no other biological experiment, in one experiment you come to a conclusion this strong and solid. So, I want to keep saying this again and again to emphasize the power of the genetic methods. So, this is how the genes are discovered. After that you can do whatever you want, you can crystallize, solve the structure, find out what happens in the active site or whether it binds to a ligand or whatever, all that you can do. But to get to the molecule, someone has to identify this molecule is required for this function, right, and that usually starts with genetics. So, that is the power of genetics here. One experiment and then you come to the conclusion this gene is required for this. Then Lin15, you find all of them getting induced, nobody is staying in the uninduced default state, indicating that Lin15 is required for that uninduced fate, which we thought as the default state. Then sometimes you get dominant alleles. So, in this screen how we would have identified this? No, in this screen, you tell me in this screen where I would have seen. To help you, I will go back to that. Yeah, you do not need to think that hard. In F1 you will have the phenotype, okay. Let 60 dominant allele was again all induced fate, but we do not make conclusion here, because we do not know what normally let 60 is doing during vulval development. So, when it is a dominant allele, it is not easy to conclude at this stage. So, the next slide tells you why. Okay, inferring functions of dominant mutations are not easy. This will require further experiments, because the dominant mutation could have risen from any one of these 3 types. It may be hypermorphic, meaning normally expressed, now you are actually expressing lot more, you are probably producing the gene product multiple doses or at least more than the normal. So, therefore, normal what it does you can't immediately say, or it could be neomorphic, meaning it may be a new function. Normally this protein does not have that at all, like many of these neurodegeneration phenotypes, the cause is actually a new function of the protein. Like for example, if you take a protein like superoxide dismutase, normally it is a superoxide dismutase, but a mutant version means it aggregates. That aggregation is a neomorph, it is a new phenotype. So, you do not know whether the dominant allele was a hypermorphic allele or is it a neomorphic, you do not know right at that step, or it could be antimorphic, it is opposing what it normally does. Normally probably it suppresses something, but the mutant version is actually not 0, instead it is promoting that. So, that is antimorphic. So, the dominant allele could be any one of these 3, and unless otherwise you identify which one of these 3, you can't immediately conclude from the observed phenotype. So, usually to settle clearly on such things like what is the wild type function of that gene, you need to get a recessive loss of function alleles as well. Okay. So, we will stop here. So, in the next class we are going to continue on this. Okay, I found mutants, I have done complementation, now I have one allele for a given gene, let us say for our Lin26. Now, where is Lin26, on which chromosome and what protein it encodes. Okay. So, we will try to focus on in the next class. Then we will move on to how do we handle dominant alleles. Okay.